Hi class, we meet again on week 6, in which based on our lesson plan, today we will learn on the third stage of aerobic respiration, which is oxidative phosphorylation, and we will also learn about another type of respiration, which is anaerobic respiration, which divided into two types of fermentation, which are alcohol fermentation and also lactic acid fermentation. So the first one, the last stage of glycolysis is referred to oxidative phosphorylation, in which this oxidative phosphorylation is divided into three main processes, which are electron transport chain, chemiosmosis, and also ATP synthesis. Now, first we're gonna look on the electron transport chain. So, in electron transport chain, it consists of four complexes of integral membrane protein, which also known as protein complex 1, 2, 3, and 4. And the name of protein complex 1 is NADH dehydrogenase complex. Protein complex 2, succinate dehydrogenase complex, protein complex 3, cytochrome C reductase complex, and we have the last protein complex number 4, which is cytochrome C oxidase. And this respiratory change also consists of two mobile carrier or two freely diffusible molecules, which known as ubiquinone or coenzyme Q, and also cytochrome C, in which these two mobile carrier function in shuttling the electron from one complex from another complex. So remember, electron transport chains do not generate ATP directly, in which the movement of the electron across or along the electron transport chain, which consisting of the mobile carrier and also protein complexes, later will contribute to the process of chemiosmosis and ATP synthesis, in which throughout this process later we will generate ATP. So this is the structure of electron transport chain and chemiosmosis. Thus, all of these make up the oxidative phosphorylation. Now, first we look at the electron transport chain over here. So we have four complex protein complexes: protein complex 1, 2, 3, and 4, and it also consists of two mobile carrier, which is, as you can see here, we have ubiquinone or coenzyme Q, and the second mobile carrier is cytochrome C. Okay, and all of these are located inside the inner mitochondrial membrane. Okay, so it's the inner mitochondrial membrane is between the intermembrane space and also the mitochondrial matrix. Okay, so meanwhile, chemiosmosis and ATP synthesis consists of one complex which known as ATP synthase, which later responsible to generate the ATP. And these two processes, including this one, thus make up the oxidative phosphorylation. So, let's look on the function of NADH and also FADH2. So, we have NADH over here and FADH2, which are extracted from the food during the glycolysis and also Krebs cycle process, and we transfer it into the ETC or electron transport gel that build into the inner mitochondrial membrane. So the red arrow here, as you can see, the red arrow trace here is referred to the transport of the electron, which finally pass to the oxygens at the downhill end of the chain. Therefore, oxygen is the final electron acceptor, and thus this hydrogen and oxygen later will produce water molecule. Okay, and next, these two mobile carrier, which is ubiquinone or coenzyme Q and cytochrome C, move very rapidly, transferring, transporting, or ferrying the electrons between large complexes to another large complexes. So for ubiquinone, it transfer electrons from protein complex 1 to the second complex and then or the third complex. Meanwhile, cytochrome C transfer the electron from protein complex 3 to protein complex number 4, and this thus give the electron to the final electron acceptor, which is referred to the oxygen. Meanwhile, protein complexes 1, 2, and 3, okay, which is actually is referred to 1. We have protein complex 2 here. It does not show in this figure. And then protein complex 3 and 4 accept and donate the electron that they got it from the NADH and also FADH2. At the same time, they pump the proton. The proton here is referred to the hydrogen ion, in which they pump the hydrogen ion from the mitochondrial matrix into the intermembrane space. So it does across the inner mitochondrial membrane through the protein complexes. So FADH2 over here deposit its electron to ubiquinone, okay, or coenzyme Q, and so result in fewer protons that being pumped into the intermembrane space than occur in NADH. So we can say that NADH is the major the major proton zonet for the process of ETC. And the chemical energy originally harvested from food is transformed into proton motive force, in which it is the chemical energy here is referred to the hydrogen ion, in which the gradient of hydrogen ion from high concentration gradient to low concentration gradient will transform into proton motive force as it across through the ATP synthase. So, what is chemiosmosis? Electron transport change and the synthesis of ATP are coupled through proton gradient across the inner mitochondrial membrane in eukaryotes. So, what happened during chemiosmosis? The proton, or referred to the hydrogen ions, flow back down the concentration gradient. So means from high concentration to low concentration gradient, from low concentration area via ATP synthase, which is built into the membrane nearby. So this ATP synthase that embedded in the inner mitochondrial membrane harness the proton motive force to phosphorylate ADP, thus forming ATP. So together, electron transport chain and chemiosmosis with ATP synthesis will make up the oxidative phosphorylation, thus produce a lot of ATP. So let's see more clear on how does the ATP synthesis and chemiosmosis occur. So this is the protein complex here is the structure of ATP synthase that embedded in the inner mitochondrial membrane, which actually responsible to make the ATP from ADP and also phosphate. So ATP use the energy of an existing proton gradient to power the ATP synthase. So the proton motive force that formulate from the hydrogen ion across the inner mitochondrial membrane is the one that is the one that responsible to power the ATP synthase. So let's look on the structure of ATP synthase here. We have rotor. In this one is referred to the stator, the rod inside, and the last knob of ATP synthase, in which this rotor within the membrane will spin clockwise when hydrogen flow pass down its concentration gradient, and then the stator over here anchor in the membrane that holds the knob stationary. It helps to hold this knob stationary so that it doesn't move at all, and the rod inside here is extending from the rotor to the knob, also being spin when hydrogen ion flow pass down its concentration gradient, and thus the rotating of the rod later will activate the catalytic sites of the knob. And three catalytic sites in the stationary knob over here, meaning to say the knob does not rotate at all, will join the inorganic phosphate to ADP, thus produce the ATP molecules. So, this is the whole process of the oxidative phosphorylation. So I will repeat. NADH and FADH2 will donate hydrogens and also electrons to protein complexes and mobile carrier, in which all the protein complexes, we have 4 protein complexes, 1, 2, 3, and 4, and these protein complexes will pass the hydrogen ion from mitochondrial membrane to intermembrane space. At the same time, it donates the electron that it gets from NADH and FADH2 to another protein complex, and this protein complex, another protein complex, will accept and transfer it to the next protein complex. And these two mobile carrier, which is ubiquinone and also cytochrome C, responsible to ferrying the electron or transport the electron from one protein complexes to another protein complexes. And the last electron acceptor or the final electron acceptor is referred to the oxygen, in which this oxygen later will accept the electron and thus forming hydrogen molecules from hydrogen ion and also oxygen molecules. So meanwhile, the high concentrations of hydrogen ion inside the intermembrane space will goes down its concentration gradient and thus producing a proton motive force. This proton motive force later will help to rotate the ATP synthase, thus activating the catalytic site of ATP synthase, and this catalytic site later will phosphorylate the ADP and also inorganic phosphate to produce ATP molecules. So this process is also known as chemiosmosis. ETC and chemiosmosis thus make up the oxidative phosphorylation process. So in conclusion, water molecules NADH, FADH, water molecules NAD, and NAD and FAD plus. It's not NADH and FADH2, yeah, it's correction. NAD and FAD plus and ATP are produced, and it occurs across the inner mitochondrial membrane. So the final electron acceptor is referred to the oxygen, in which the electron they got it from NADH and FADH2. So this is the video of how does the oxidative phosphorylation occur. I will attach the link later of the video. So where do we get the NADH actually? So most of the NADH in electron transport chain are produced from Krebs cycle and also glycolysis actually in the mitochondrial membrane. So mitochondrial matrix, so direct accessible to electron transposition. Meanwhile, the inner mitochondrial membrane is not permeable to NADH from cytosol. So NADH from the cytosol is the one that being produced during the glycolysis process. So therefore, the electron from NADH is transferred to ETC through shuttle system, and there are two types of shuttle mechanism. First, we have mallet and aspartate shuttle, which is used liver, kidney, and also heart, um, in which the electron from NADH, which is referred to the cytosol, transfer to NAD plus in the matrix of mitochondrial. The second shuttle mechanism is glycerol phosphate shuttle that used in skeletal muscle and also brain, in which the electron from the NADH inside the cytosol will transfer to FAD plus in matrix of mitochondria. So what is actually the role or the functions of NAD plus and FAD? So NAD is actually coenzyme and an electron carrier, and it acts as an electron acceptor in glycolysis and also Krebs cycle through dehydrogenation process, and is then reduced to NADH. So excuse me. So when it is reduced to NADH, it is utilized as an electron donor during oxidative phosphorylation. Meanwhile, for FAD is the oxidized form, and its reduced form is FADH2, and it is also a coenzyme, same like NAD+, and it comprises of a phosphorylated vitamin B2, or known as riboflavin molecule, that link to the nucleotides adenine monophosphate, and it functions as a hydrogen acceptor also in dehydrogenation reactions, so it thus being reduced to FADH2. So in summary, 36 or 38 ATP is being produced through cellular respiration or aerobic respiration. 2 ATP and 2 ATP from Krebs cycle, so 4 ATP. 2 NADH produced from glycolysis will go to the oxidative phosphorylation and thus help to make up 3 or 5 ATP. 2 NADH produced from the formation of acetyl-CoA will go to the oxidative phosphorylation, thus help to produce 5 ATP. 6 NADH and 2 FADH2 molecules from the Krebs cycle will also go to the oxidative phosphorylations, and thus it helps to produce 15 ATP to 5 ATP, and the total of it does make up 36 or 38 ATP. So here the review of the cellular respiration, much more clearer. 2 ATP from glycolysis, 2 ATP from acetyl-CoA, in which we will get it from NADH later, 2 ATP from Krebs cycles, and also about 34 ATP from electron transport change and chemiosmosis process. So in cellular respirations, 3 most important details that you need to know. The first, you have to know what is the molecular inputs and the molecular outputs from each stages. How many ATP molecules are being produced from each stage, and how many NAD plus and FADH molecules are reduced at each stages. So this is the video of the cellular respiration. I will send you the link later. And next, we're gonna look on the second type of respiration, which is also known as anaerobic respiration. So under anaerobic respiration, the absence of oxygen, which is referred to the absence of oxygen, the pyruvic acid, or known as pyruvate, can be route by the organism into lactic acid fermentation and also alcohol fermentation. So this fermentation occurring without oxygen, and it is the partial degradation of organic molecule, which is referred to the pyruvate. So it produces ATP. How? By substrate level phosphorylation. By substrate level phosphorylation through the process of glycolysis. So fermentation is actually refer into two anaerobic respiration is actually consisting on two stages. We have glycolysis, and then the second stages is fermentation. And this fermentation is divided into two types: alcohol fermentation or lactic acid fermentation. That means glycolysis to alcohol fermentation or glycolysis to lactic acid fermentation. So later, the glycolysis will produce NADH, and this NADH will use by fermentation. Thus, the fermentation will generate NAD plus, and only glycolysis can make up the ATP. So, this is the overall view of how does the anaerobic respiration occur, in which the glucose will undergo the glycolysis process, thus produce pyruvate, and if there is an oxygen present, this pyruvate will undergo formations of acetyl-CoA, citric acid cycle, and also oxidative phosphorylation. But if there is an absence of oxygen, this pyruvate that produced from the glycolysis will go through the process of fermentation, either ethanol or lactic acid fermentation. So two organisms that use anaerobic respiration to produce ATP, which is facultative anaerobic and also obligate anaerobic. So organisms, facultative anaerobes is an organism that makes ATP with the presence of oxygen, and it's capable to switch it to fermentation when there is no oxygen. Meanwhile, obligate anaerobes is the organism that will die in the presence of oxygen because oxygen is toxic to this type of organism, and instead they use sulfate as final electron acceptor. So let's look on the two types of fermentation. Remember, fermentation consists of glycolysis plus reaction that regenerate NAD plus, which can be reused by glycolysis. So remember the main function of the main functions of fermentation is to regenerate NAD plus. Okay, I repeat, the main functions of fermentation is to regenerate NAD plus. And the two common types of fermentation are lactic acid fermentation and alcohol fermentation. So, how does the alcohol fermentation occur? First, the glucose will produce 2 ATP, the net gain of 2 ATP through glycolysis process, and this glycolysis process will produce 2 molecules of pyruvate. 2 molecule pyruvate will go to alcohol fermentation by removing 2 molecules of carbon dioxide and produce 2 molecules of acetaldehyde. And then this acetaldehyde will help to regenerate and 2 molecules of NAD plus from 2 molecules of NADH, which later will being NAD plus, which later being used by glycolysis process, and thus the acetaldehyde forming 2 molecules of ethanol. So this alcohol fermentation is used by many bacteria and also yeast, and um, application it's being applied in bakery products such as cake and bread, beer, wine, and also local foods such as tempeh, tapai, and also budu. The second type of lactic acid is lactic acid fermentation, in which, I repeat again, glucose will produce a glucose through glycolysis process will produce 2 ATP and also 2 molecules on pyruvate, in which these 2 molecules pyruvate will enter the lactic acid fermentation by regenerate 2 molecules of NAD plus, in which these 2 molecules of NAD plus will being used by the glycolysis process, and at the end these 2 molecules of pyruvate will produce 2 molecules of lactic acid, and this lactic acid fermentation is used by lactobacillus bacteria that you can find it in the cheese and also yogurt, and also being used by human for the quick generations of ATP during intense activities such as running, and also it present of in lactate muscle when you feel muscle cramp and also fatigue. So this is the differences between lactic acid fermentation and alcohol fermentation. First, it occur in which types of organism, and then alcohol fermentation does involve 2 steps. Okay, from pyruvate to acetaldehyde to ethanol, and it produce carbon dioxide. Meanwhile, for lactic acid fermentation, it's only involve 1 step from pyruvate to lactic acid fermentation, and it does not produce any carbon dioxide. And the final product of alcohol fermentations are 2 molecules of ethanols and 2 molecules of carbon dioxide. Meanwhile, lactic acid fermentation produce 2 molecules of lactic acid, and alcohol fermentation it's being applied by the yeast that used in brewing, winemaking, and also baking. Meanwhile, lactic acid fermentation is being this process is being applied by some fungi and also bacteria used to make cheese and also yogurt. Therefore, in conclusion, we can say that the 3 fates of pyruvate, or the 3 last destination of pyruvate that after being produced from glycolysis, which is the pyruvate will be used in anaerobic respiration such as lactic acid fermentation, and it also being used in an alcoholic fermentation, and it also the pyruvate also being used for aerobic respiration through the process of formations of acetyl CoA. So, glycolysis and citric acid cycle connect to many other metabolic pathway, in which that it is in which these 2 process are the major intersections to various metabolism or metabolic pathway. First, for protein must be digested to amino acid, in which this amino acid later will be used in the process of glycolysis and also citric acid cycle or Krebs cycle. Meanwhile, fats later will digested to glycerol and also fatty acid, in which this glycerol will be used in the glycolysis process, while the fatty acid later will be used during the formations of acetyl CoA. And the fatty acid are broken down by beta oxidation and yield acetyl CoA, in which acetyl CoA, remember acetyl CoA is the one that the molecules that are responsible to start the Krebs cycle process, and an oxidized gram of fat produces more than twice as much as ATP as an oxidized gram of carbohydrate. So this is the how does the proteins and also fats and also the carbohydrates being used for the process of aerobic respiration. The protein will digested to become amino acid through the process of deamination before it goes through glycolysis, formation of acetyl-CoA and citric acid cycle, and the fat. Meanwhile, the fat be digested into glycerol and fatty acid, in which these two type of fats will used in the process of glycolysis and also acetyl-CoA. Therefore, in conclusion, aerobic respiration include three stages: glycolysis, Krebs cycle, and oxidative phosphorylation. Meanwhile, anaerobic respiration um, only involve two stages: glycolysis and also fermentation, in which we have two types of fermentation: lactic acid fermentation and alcohol fermentation. Remember, aerobic respiration produces 36 up to 38 ATP. Meanwhile, the anaerobic respiration only produces two molecules of ATP. So I think that's all for today, class. Thank you. See you next week.